Let me briefly talk about the overview of cellular respiration in a big picture. Of course, it starts from the glycolysis, simply meaning the sugar splitting. The glyco you see up here has a prefix of sugar, and the lysis is splitting. So it's a sugar splitting. So we start with the glucose, and then we break it into two pyruvate, or a pair of pyruvate. Glucose has 6 carbons and pyruvate has 3 carbons. In the process, we're gonna make 2 net ATP. It starts with the energy investment of 2 ATP, and then energy payoff, we get 4 ATP. So the net gain of ATP in the glycolysis is 2 ATP. But that's not all we make. We also make NADH, 2 NADH, and they are the potential ATP to be processed in much later stage. You see oxidative phosphorylation. Okay, it comprises of electron transport chain and chemiosmosis via the ATP synthesis. ATP synthase. Alright, so we make the 2 NADH, and in this process, we also create the water, and again we make pyruvate, and then it goes through the mitochondria. So remember that the glycolysis happens in the cytoplasm or cytosol, and then once the oxygen is present, then it undergoes the aerobic cellular respiration. That means it enters through the mitochondrial membrane, and the pyruvates are oxidized here. In this process, which is not shown here, the carbon dioxide is released. Also, we create more NADH. And then once we convert the pyruvate to acetyl coenzyme, it enters into the citric acid cycle, or the Krebs cycle, named after the guy who researched this. And then we make ATP as well as NADH and FADH, yeah, and more FADH and FADH2 actually. Okay, they act as a electron carriers to be processed in the final stage here. So what happens is these electrons we see, or electrons gained through the reduction, will be cascading down the electron transport chain. And these electron transport chains are a series of proteins that transfer the electrons, creating the proton gradient on the intermembrane space, and using that gradient by simple diffusion, we're gonna create the ATP by rotating the ATP synthase. So we're gonna talk more in detail how the oxidative phosphorylation works, but in the nutshell, we start with the glucose and then end up with a lot of ATP when oxygen is present. Now, in case the oxygen is not present, it undergoes through the fermentation, which I'm going to talk much later.